Hello, everyone. Welcome to my MHRE lecture. My name is Spencer Ames. I'm a master's student in the Department of Immunology. I'm in Dr. Meghna Zad's lab, and today I'm going to present on the topic of infant feeding. And I'm trying to introduce you all to the very complex question, how do early life experiences shape lifelong health? So first, before I really dive into my presentation, I want to give a little about me, introduce myself to you all. So I am born and raised in Calgary, and I went to the University of Calgary to complete my undergraduate degree in cellular, molecular, and microbial biology. And throughout my degree, I didn't really know what I wanted to do afterwards. I was thinking, okay, do I want to go to medical school? Do I want to do something research? Do I want to go a completely different direction? So during my time, I tried to do a lot of different activities, and mainly my research activities. I tried to do it in a few different fields. So I did some Parkinson's disease research. I did some theoretical research on muscle kinetics, and I spent quite a bit of time at the Alberta Children's Hospital looking to see if the introduction of a humanoid robot could help reduce anxiety and fear for kids that came to visit the hospital. So I'm doing all of these activities to try and determine, okay, what do I want to do next? But when I'm doing all these activities and through different courses, I find I liked a lot of different fields of research. And that made it difficult for me to choose a direction for my master's, which I'm in now. And I ended up finding my supervisor, Dr. Meghna Zad's lab, and reading about her research, the Thrive Discovery Lab's research. I found an area of research that's very multidisciplinary. I'm able to learn new things about a lot of different fields, a lot of different areas. And I think that's very unique for my master's. And I wanted to share that unique experience with you all and maybe shine a light on some different types of research in health sciences. So for my presentation today, I'm going to be talking about three main topics. The first, I'm going to introduce multidisciplinary research and DOHAD, or the developmental origins of health and disease. The objective for this first section of my presentation is to allow you all to identify different areas of multidisciplinary research and to be able to identify and discuss DOHAD research. For the second chapter of my presentation, I'm going to be discussing infant nutrition research. And I really want you all to be able to discuss the significance of infant nutrition research, as well as discuss the roles of different research fields when studying and answering questions in infant nutrition. For the final chapter in the last half of the lecture today, I'm going to be leading a workshop on decision-making and evaluating interests for potential career options. So I want to warn you guys right now that for this exercise, I will be breaking you all up into breakout rooms, groups of 4 to 6, where I hope you all do a little bit of discussion and discuss some career interests. We'll get to that later, but I wanted to give the little warning right now. So to start, I want to ask and answer, what is multidisciplinary research? I have a very simple definition right here. Multidisciplinary research draws from 2 or more research disciplines in order to gain a more well-developed perspective or discover something new. When we look at fields like biochemistry, it's fairly simple. We have biology, chemistry, and how they interplay is very important for that field. Something like neuroscience can use physiology, physics, computer science to answer research questions. And I have in bold here, developmental origins of health and disease, or DOHAD. And this is not necessarily a research field, but it's an approach to research that is definitely attempts to account for various facts and knowledge of different fields and topics to answer complex problems. So what exactly is DOHAD? DOHAD is an approach to medical research based on the thought that exposures during critical periods of development and growth may have significant consequences on an individual's short and long-term health. So this is kind of an approach developed, or it's been known, but it was mainly developed from epidemiological studies done by Dr. David Barker, where he connected fetal undernutrition to disproportionate fetal growth and coronary heart disease later on in life. So studied exposures in DOHAD often include things like nutrition, looking how infants are being fed, and environmental exposures. What is around these infants that is causing these changes in short and long-term health? So these exposures can result in permanent changes to different biological processes in infants that affect health and are important to be taking a look at. So I have here just a very standard and simple example. An example of environmental exposure is secondhand smoking exposures. So for an individual in utero or in his or her mom or during childhood, with secondhand smoking exposure, that individual in childhood has a greater likelihood to have upper and lower respiratory infections, to develop asthma, ADHD, emotional problems, and hearing loss. So this is a clear example of an environmental exposure linked to some longer term consequences. Nutrition is also involved in these processes, as well as something like obesity, diabetes. How an infant is being fed has a great association in the risk of those diseases as well. So I want to also mention that it's never like being exposed to secondhand smoking leads to this. It's always a collection. It's an interplay between a lot of different factors, a lot of different exposures, a lot of things that are environmental exposures, and a lot of things that aren't. So, for example, some other factors are also linked to growth and development. There's so many factors, but here are just a few examples. Some early life factors, maternal factors, and infant factors. This includes things like birth mode and the number of siblings. It's been shown that depending on your birth mode, whether that be like C-section or other, and the number of siblings can impact the amount of microbes and an individual can be exposed to, which can impact infant development. Furthermore, something like maternal age is associated with the risk of a variety of different diseases, such as diabetes and increased likelihood for hypertension. Something like gestational age, that's something where something like early term infants have been associated with having slower neurodevelopment than those born at full term, which can obviously affect growth and development of infants. So overall, I just want to highlight that there's many things going on that dictate growth and development. Therefore, when we are trying to answer DOHAD questions, is this risk have an association with this disease? We need to take those factors into account. Hold on, I just have a question here. Is this type of research related to the social determinants of health? Absolutely, 100%. Social determinants of health is 100% relevant to this area of research. There can be something like socioeconomic status is often talked about and accounted for in this type of research. Different social determinants everywhere in the world, different cultures trying to be accounted for. Definitely a very relevant factor for this area of research. So overall, I also wanted to mention, what is the goal? What do I think the goal of DOHAD research is? What is the significance of this? How can this affect our health? Well, in my mind, understanding infant development and growth can help us understand how to provide the best start to life. I have this figure here, and it's showing brain function and behavioral competence trajectories on the y-axis, but overall, I want you to just think of overall development of an infant. And then we have over time, so prenatal, birth, early childhood age, we just have over time. We have green as optimal growth and development, red, not so optimal, and blue is kind of a recovery. But when I'm thinking about DOHAD research, I'm thinking about, for example, an infant that has been introduced, an individual that has been introduced or exposed to secondhand smoking in utero. When that infant's born, they might have a greater risk at developing something like asthma. So that might put them on this red line. If we do more research on what that means, what are the mechanisms of secondhand smoking that are causing that infant to be more likely to develop asthma? If we can understand those factors, we might be able to develop treatments to get that infant or that individual off of this different trajectory and improve or lessen the likelihood of them developing something like asthma. So this is obviously an idealized and kind of simplified approach. It's not accurate in every instance, but I think this kind of highlights the more we understand about infant development, the more we can kind of treat these things. As we start detecting something like asthma or another disease, we can try and make an intervention to correct for those things. So where does my research project, that's a lot of talk about DOHAD. Where does my project, what am I doing? So I'm doing a very small part of this overall picture, very complex, a lot of factors associated. I'm looking at the association between infant feeding practices, something like human milk feeding duration, exclusive human milk feeding duration, and method of human milk feeding duration. I'm looking to see how that influences early life immune system development. So my project's really focusing on the nutritional aspects of DOHAD and infant development. Therefore, for the remaining part of my project, I'm going to focus on proper nutrition, and I'm going to discuss different multidisciplinary approaches that I'm using in my project going ahead. We also talked a lot about the long-term consequences of some of these environmental exposures. I want to zone in on some immediate and direct impacts of early life exposures and nutrition. Inadequate infant nutrition is an issue. 35% of under 5 deaths are due to the presence of undernutrition or are linked with some sort of factor associated with undernutrition. This is definitely a worldwide issue, as undernutrition, even though it might not lead to death, it could lead to an impairment in growth, stunting, impaired intellectual performance, capacity for work. I feel like in a place like North America, it's not as discussed as some other places. But when we hear things about the infant formula shortage going on in the US right now, it's definitely a very relevant issue to be discussed. Some infant or mothers are being rushed back to work, not able to breastfeed infants for an appropriate time or amount. Definitely a very relevant topic. So I want to focus on human breast milk now. It's considered the gold standard for infant nutrition. It contains components that can satisfy nutritional requirements, fats, lipids, carbohydrates, those types of things. And it also contains components that make it associated with protection against infections, increases in intelligence, and reduced risk of things like obesity. There's a ton of benefits for sure. One epidemiological study found that scaling up breastfeeding so that infants are all receiving an adequate amount of breast milk that can satisfy their nutritional requirements, it can save an estimated 823,000 child deaths every year. Obviously, this is a mathematical model, but it just goes to show how many infants are not being adequately fed and how important and useful a tool like breastfeeding can be. I'm getting a question here also saying, do you know if breastfeeding is directly related to an increase in life expectancy? Personally, I haven't read anything looking at the association between breastfeeding and overall life expectancy. Potentially, I cannot answer that question, unfortunately. So moving on, what makes human breast milk special? I mentioned it has all of these components, which I'll talk about later, but it also has some unique abilities that are like very personalized. It's been talked about as being that personalized nutrition that can match the nutritional needs of an infant. If an infant needs more of something, it can provide that. It can, the interaction between a mother and an infant can signal for the mammary gland to produce different compositions of human milk. Very cool. It's also been referred to as a personalized medicine, as it can deliver immunity while an infant immune system develops. And overall, human breast milk research, human breast milk itself is quite, it's not very well studied. There's like a common saying in a lot of breast milk researchers. They say that strawberries are studied more than human breast milk. So it goes to show there's a lot of cool capabilities, not very well understood. So there's a bunch of different questions, and I just have 4 here, and I'm going to focus on one of these, but I'll just mention all of these questions. We have, how does human milk assist and aid in the development of infant immune systems? How does human milk composition change according to the needs of an infant? Why does the composition of human milk vary around the world? What impact does this have? What are the major differences between human breast milk and formula? So there's tons of unanswered questions that are being investigated right now. But as my project is looking at infant immune system development, I want to focus on this first one here. So here I have that question. And in the next steps for my presentation, I want to look at these 4 different research fields and how they're all interplaying and having a role in answering this question. It's definitely not one that we can look at as just having one answer. There's not one approach to answer this question. There's a variety of different approaches. So starting with microbiology. A lot of microbiologists look at human milk and the microbes and fungi that are transferred from mother to infant via human breast milk. I do want to kind of take a little step back, and I know there were quite a few microbiologists or undergrad students in microbiology, but why is this important? Why is the transfer of microbes and fungi important? Well, according to the hygiene hypothesis, exposures to particular viruses, bacteria, fungi, and parasites lead to the early maturation of the immune system and reduced incidence of asthma and allergy, as well as other diseases. So when we have the transfer, it's providing some sort of, here we go. So yeah, the presence and transfer of human breast milk microbes such as bifidobacteriae and lactobacilli. One example of something it can do is it interacts with receptors in our gut to enhance the gut barrier. So it can influence the development of the gut, the oral microbiome of an infant, and it can allow microbes to, have better protection from things like invading bacteria. As we show here, when we have these commensal bacteria that can line our gut microbes, there isn't any really room for these invading bacteria to go and cause some sort of pathology within infants. Furthermore, microbes also take some human milk components and digest them. It serves as kind of a food or a prebiotic for these microbes. And then these microbes can release some other substrates that are also shown to kind of mature the intestinal barrier. I have another question here. Would you suggest introduction of different fermented foods from 6 months onward? Definitely, this is a good question. There's a lot of talk about, even in different formula, they have like different probiotics being added to different formulas. Even like going forward, things like yogurt have probiotics, prebiotics. Would I suggest the introduction? At 6 months and onward, in my mind, or based on my knowledge, things like the gut microbiome are fairly well colonized at this point. So an introduction of different fermented foods, will it have a positive impact? I'm not sure. It might be something like after someone takes antibiotics and maybe the gut microbiome is depleted, maybe the introduction of some fermented foods with commensal bacteria that are shown to be good for infant health, that could be an option. But it's a tough question. Would I suggest It depends. But yeah, so human milk microbes being passed from a mother to an infant have a very important role in aiding the development of infant immune systems. Moving on to nutrition, I'm going to list off a bunch of other factors that are in human milk that are important for infant immune systems. First off, human milk contains a bunch of different hormones. An example of one is FGF21, fibroblast growth factor 21. That's a factor that is associated, it's an insulin sensitizer, first off. It's associated with regulating metabolism in infants, and it's been shown to have a protective effect against diet-induced obesity. This is something that is still being very heavily researched, but as obesity is something that has can lead to low levels of inflammation, it's definitely an important factor and can impact infant immune systems. Human breast milk also contains bioactive enzymes such as lactoferrin, which functions by kind of taking iron in the blood and chelating it, hiding it from different bacteria that could use that as and have a pathogenic effect. Essentially hides, iron hides iron and has that impact. So again, that can be important to avoiding potentially dangerous microbes. Breast milk also contains human milk oligosaccharides, which I mentioned. Well, I referred to them earlier because those oligosaccharides are the food for gut microbiota. Those are digested by a lot of the bacteria there and have different functions in enhancing gut barriers. Different micronutrients present, such as vitamin E, provide antioxidant protection, and something like zinc is important for enzyme function. So again, it could have a role in different enzymes or immune enzymes and proteins. It could assist with those, the proper development of those proteins. Different growth factors, such as epidermal growth factor, also are associated with maturing and healing intestinal barriers. So definitely there's many components that are associated with the immune system within human milk. Continuing on with a similar theme, I want to kind of switch more to the immunologist side. Now, bringing in immunology, human milk, and this is something that is fairly well known in health science, it does contain immunoglobulins or antibodies. These immunoglobulins can provide a passive immunity to developing infants. When I say passive immunity, I mean that when those antibodies are present, the maternal antibodies within an infant, when they're passed on, they might be able to react with different microbes that an infant hasn't made its own antibodies for. Something that is talked about a lot is it's similar to receiving monoclonal antibodies as a treatment. I know that was discussed a little bit around COVID treatments. Some people were receiving monoclonal antibodies to help stimulate an immune response. It functions in a very similar way. Once an infant receives some of these antibodies, it can have protection, or at least it can stimulate immune responses to different pathogens that the mother has already developed an immune response for. Two other components or two other subsets in human milk are immune cells and cytokines. And there's a bunch of different functions these can have when they're passed from a mom to an infant. But overall, I like to look at these like things like macrophages and dendritic cells, as well as different cytokines. They're tutors for the infant immune system. They can share signals with developing cells within an infant that can stimulate proliferation, differentiation into a bunch of different things, cause many different reactions. So I look at overall immune cells and cytokines, they're kind of tutors. They're saying, okay, here's some antigens, here's some growth factors, let's get things going. And overall, this is I guess a fairly complex graph. This is showing everything that I just mentioned related to things within human milk and how that relates to infant immune system development. We see our immunoglobulins here. more immunoglobulins, microbiota having a role in developing immune cells. These immune cells leading to the inhibition of inflammation. We have human milk oligosaccharides being a substrate for microbiota to eat. We have enzymes, lactoferrin, they're providing protection from pathogens. Inflammation, definitely a process regulated by human milk. So overall, this is kind of a figure showing what I've mentioned so far. I have another question here. If a lactating mom got COVID, but not the actual COVID, will the antibodies, got the COVID vaccine, but not the actual COVID, will the antibodies also transfer to the baby? In theory, absolutely. Especially if the baby did have, let's say early on in life, where the intestinal barrier is not fully developed, that baby might actually have like or get protection that's pretty strong. I'm not 100% familiar with like some specific studies that have looked at this, but it's definitely been a topic of discussion in human milk research. So my answer, yes, but I also don't want to talk too much about it because I don't know absolutely everything about COVID for sure. So moving on to community health sciences and epidemiology. In the last little bit here, I'll just talk about taking a step back and looking at overall populations. I just want to quickly define population level research in community health sciences and epidemiology as the study of distribution and determinants of disease or health outcomes in specified populations, and applying this study to help with health problems. So where all these studies are looking at overall health outcomes, what this means, should we be adjusting things like health policy? Should we change different recommendations for human milk feeding? And what are the health outcomes here? So taking a step back and looking at a population level to this question can suggest how human milk is overall affecting the end results, the development of immune systems. What are the end results? It's taking a look at the overall, is there disease, is there not disease? What is the percentage of this? What is the percentage of that? Is it likely, is it not likely? It's taking a bigger step back. It's moving from the lab bench to population. For this, I have a couple questions. Are different feeding practices associated with different health outcomes? How should we be providing nutrition? Those are some questions that this population level research is taking a look at. An example of something that has been investigated in this way is this statement right here, or this recommendation. The World Health Organization recommends exclusive human breast milk feeding for 6 months. What's that based on? Well, they take a look at different population cohorts. They look at mothers that have fed for this long, mothers that have fed for this long. What else have those mothers had? Are those mothers smokers? They look at all of these different factors, and then they pump out, or they do an investigation. They develop models, and they found that infants that are exclusively receiving human breast milk for 6 months, when compared to those who are breastfed for shorter periods or not at all, have a lesser risk of contracting GI tract or respiratory infections. So overall, coming back to this question, there's a great interplay between these fields and others. I also have, I've added genetics, pharmacy, infectious diseases. These are also factors that are impacting immune system development. So again, this just highlights how complex this question is to answer. You got to break it down into smaller subcomponents to get some answers. But it also highlights just how interesting this research is. As a researcher in this field, I get to learn things about immunology, medication, different disease, different techniques for investigating population. I get to have all these different components to answer a small subset of a very cool research field that can provide some benefits for health of infants and provide a good start to life. Bringing it back to here. So one other thing that I'll mention is, again, very complex, a lot of different fields. But finding this field was very difficult for me. I did a lot of different looking around, and I'm not sure if you all are in similar shoes right now looking for potential grad school. I'm sure some of you guys are going to grad school in the fall, but finding a research field that you're passionate about, thinking of a career that you're passionate about, it's always a tough choice. And that's what really kind of inspired me to form this workshop on career evaluation and decision making. I have Toby sent out an Excel sheet earlier this week, and I'll share it. I'll share my screen and show and kind of walk you guys through the Excel workshop exercise. But I made this with the hope that it's sharing a resource that you all can use for evaluating different decisions. Not only can this be used for career evaluations, but you can adjust this Excel sheet and ask yourself different questions. Okay, you can change it to fields of research. You can change it to evaluating decisions in relationships and such. I find this is a very helpful technique. So I'll ask now. I'll share my screen here. Can everyone see my Excel sheet? Toby, can you see it? So I'll ask everyone, if they haven't already, pull up this worksheet, get prepared. I'll walk through it quickly, and then I'll ask Toby to divide you all up into groups. But what I've done in this Excel sheet, I have two sheets, exercise and then the group assignments. So the first thing, once you're broken up into your breakout rooms, remember the number of your breakout room and go to group assignments. Go on this breakout group number, and I've assigned each breakout room 2 assigned positions. So for this first breakout room, principal investigator and genetic counselor. So you're going to take these 2 and you're going to move them over here into the assigned positions you're going to be looking at. And then along with your group members, I want you all to research those jobs. Maybe these jobs aren't of interest to you at all, but there's going to be pros and cons to every job. Maybe in this research, I really encourage everyone to do this, because maybe in this research, you're going to find out, like, maybe that is a part that I do want in my life. Maybe this has a work-life balance that I didn't really expect. Maybe it's too much work. Maybe I feel like I'm going to want more. There are definitely a lot of things to learn by investigating other jobs that maybe you're not have really even thought about. Anyways, so you're going to do research. Right here, what do I have here? Yes, so you're going to take 10 minutes per each assigned position. You're going to do research, you're going to discuss, and before the 10 minutes are up, I want each of you to color in these cells like so. Color in the cells according to the legend. So first reaction, is this position exciting to me? Totally. Okay, mark it green. And I want you to answer all of these questions here. And then in the second 10 minutes, I want you to do the same for the second position. So welcome back, everyone. I hope the exercise taught you something new. You learned something about a different position maybe, or found something that maybe you want in a future career. I wanted to bring you all back here and just talk a little bit more about it. Some of the questions, there's a section in the Excel sheet. I called it other considerations. And some of these questions vary, like based on a company, something like that. But some of the other questions are kind of hard to find an answer to online. You can do all the research you can on a specific job, but a different company just might be completely different. So I wanted to just mention something like an informational interview. I'm just going to briefly discuss it. It's the option to email or get in touch with somebody in a career you're interested in, and ask them if they'd be up for meeting. First, introduce yourself, ask if they have some time to meet, something like that. And this allows the opportunity to ask some of these questions that are hard to answer. So I wanted to talk about that. It's also a good way to find mentors that can really care about you and your development. I feel like a lot of professors or people in professional jobs do care about trainee development and helping people develop. So it never hurts to ask someone if they have some time. It can be 15 minutes, 20 minutes, less even, but I wanted to promote that a little bit right now. I also have a slide here with some additional resources, and I don't want you to like scramble to write it down or anything. I will send this information to Toby, and then he can send it out to you all. But this is also further websites, this Imagine PhD one. It evaluates your values, strengths, skills, and other things, and kind of cranks out like does this job fit you? And they're all science-related careers. Some more information related to informational interview is here as well. And this is another resource, another MyADP that focuses more on like social science careers as well. So I'll send this information out. And again, thanks for trying, and I hope you all put some effort into the exercise and listening to my lecture. Thank you very much. I'd be happy to answer any questions.